Hello, everyone. My name is Maria, and for the past 8 weeks, I've had the privilege to work with my lab PI and mentor, Dr. McDonough, in the McDonough lab on my project that is investigating microglia activation in response to different food groups in a neonatal piglet model. So for some background on my project, to better understand it, premature infants are at a high risk of infections and comorbidities that can cause neurodevelopmental delays. And there's evidence that food source may affect the development of the intestine, systemic inflammation, and neuroinflammation. However, there is not a lot of evidence to characterize this. Our collaborators at the University of Idaho are investigating the effects of infant formula and human breast milk on these outcomes. However, here at the University of Washington, we are specifically looking at neuroinflammation. And to do this, we used a neonatal piglet model since piglets have a high homology to human infants in respect to their brain growth trajectory and neuroanatomy. The main focus of my project was looking at microglia, and microglia are the resident immune cells of the central nervous system, and they're the first responders to infectious and injurious stimuli. When microglia are activated, they acquire a specific morphology, as you can see in the figure right here. To the left, you can see microglia in their surveillance state, while they have a more ramified phenotype and longer branches in comparison to the right, where you can see microglia in their activated state, they have a less ramified phenotype and shorter branches. We also see that microglia tend to proliferate in response to activation, and microglia activation is an indicator of neuroinflammation. At the University of Idaho, preliminary data was conducted in piglets that consumed human breast milk in comparison to infant breast milk. And the data showed that piglets that consumed human breast milk had higher concentrations of inflammatory and anti-inflammatory cytokines in their gastrointestinal tract. Due to their preliminary data here at the University of Washington, we are hypothesizing that piglets that are fed human breast milk will have less neuroinflammation, thus having less microglia activation than piglets that are fed infant formula. So to do this, our collaborators at the University of Idaho had 3 piglets that were assigned to each feeding group, a human milk feeding group and an infant formula feeding group for the duration of a total of 28 days. Then the piglets were euthanized and their right prefrontal cortexes were sectioned using a chirostat. Then during the duration of my project, I performed immunohistochemical staining on these slices. And this process is done by having a primary antibody that binds to a specific antigen. Then there's a secondary antibody that binds to that primary antibody. And there is a fluorescent tag that shows under a microscope. The primary antibodies that I stained for are IBA1 and Ki67. IBA1 appears green under the microscope and Ki67 appears red. IBA1 stands for microglia and Ki67 stands for proliferative cells. And we imaged using the Mariana's microscope. I also used a fluorescent dye called DAPI that directly binds to the DNA to help visualize the nuclei. Then I performed serology on my slides. Serology happens when serology allows us to quantify microglia and proliferative cells in an unbiased manner. And I did this by taking, as you can see up here, taking a specific mask of the tissue and allowing the software to take random counting boxes and phase 3 by 50 microns. Then to analyze the morphology, I used a MATLAB script that helps skeletalize the microglia and proliferative cells to give out specific outputs. Some of these outputs include average branch length and cell volume. When interpreting my results for the IBA1 positive cells, Ki67 positive cells, and the double positive cells, which again, IBA1 positive cells show microglia, Ki67 positive cells show proliferative cells, and the double positive cells show proliferative microglia. For the 2 feeding groups, human breast milk and infant formula, our results were not significant. We believe this is due to the fact that we had a small sample size of only 3 piglets per feeding group. However, when we look at the results of our morphology, we can see that for the average branch length, the human milk fed group had longer branches in comparison to the infant formula fed group. And if you can look at the figure up here, we can see that the longer branches are associated with a more ramified phenotype, thus having less proliferative cells and less activation. When we look at the average number of endpoints, we can see that the human milk-fed group had a higher number of endpoints in comparison to the infant formula-fed group. This is due to the fact that a higher a number of endpoints corresponds with more complexity to the cell, longer branch points, thus less activation and proliferation. And then finally, looking at the complexity index for the human milk-fed group in comparison to the infant formula-fed group, we can see that the human milk-fed group had a higher number on the complexity index in comparison to the infant formula-fed group. We believe this is due to the fact that the human milk-fed group had more surveillant microglia that had a more ramified phenotype that was less activated and proliferated less than the infant formula-fed group. In the future, for our pilot study, we only had 3 animals per feeding group. However, in total, we plan to have a total of 14 animals per feeding group. We also plan to explore neuroinflammation in different regions of the brain, not just the right prefrontal cortex, such as the amygdala and hippocampus. We also plan to quantify oligodendrocytes and oligodendrocyte progenitors to look at white brain matter. I would like to give a very, very special thanks to the McDonough and Weinstein labs at the University of Washington, including my lab PI and mentor, Dr. Ash McDonough, Dr. Jonathan Weinstein, and everyone in the program who has helped make this happen. I would like to thank Dr. Ellenbogen and Mrs. Ellenbogen, as well as Julie, Sylvia, and everyone. Again, I wouldn't be able to be here without you guys. Thank you.